Okay, I'm Christy Sayes. I'm formerly from Texas A&M University and now at RTI International, which is a not-for-profit research institute. So my experiences and perspective have come from Texas A&M to the last couple of years I've been at RTI. I've been developing with my team of fellow scientists two different models, one of the gut and one of the lung. And these, pardon my expression, because we just don't have really a good vocabulary right now for the researchers doing in this field, but internally we use a 2D, 3D, 4D, okay, to differentiate between what we're talking about with just our monolayers by themselves, our three-dimensional constructs, which would be either in the matrigel or collagen suspensions and deposited or used in transwells. And then when we bring in microfluidics, we usually call that the fourth dimension just in our lab, just to keep it separately for us. It may not be the best terminology for this group, but that's how we've been doing it for both particles as well as vapors, okay? So there's four major things that we've run into in the years as we develop these things and that I'll put out there, and hopefully we can talk about them some more. But the first is what Cheryl Walker brought up earlier in one of the conversations, which is just not having enough material to be able to do the assay at the end of the analysis. So a lot of the times, especially with my experience with particles, we have a lot of interferences with the assays that we choose at the endpoint. So I want to be able to validate that assay and those results with other types of tests. And in order to have enough protein or RNA or any other type of lysate, I need to have a large enough cell population to be able to have the sample and redo it and reanalyze for looking at a similar endpoint or the same endpoint but using a different methodology. So having a well that's large enough for that is very important for me in the design of our systems. And also to eliminate false positives and negatives. So this is why we work and put money into both a microfluidic system and a macrofluidic system, just to increase volume capacity of the systems. The second major point that we've run into through the years is just the sample preparation. Okay, so when we're talking about gut, we have followed in the footsteps of other people, we pre-digest all of the particles and materials that we use when we do the exposure. So it's very important not to just take the raw material as is and expose that to the cells whenever we're talking about exposure in the gut model and what's interacting with the biofilm on top and the endothelial cells underneath. We actually pre-digest our samples and put them through, this is published by lots of people, including myself, but through salival fluid, stomach fluid, and then GI fluid to be able to understand the entire system that's being exposed. Again, this is most important for particles, but for particles, this, I believe, needs to be in the discussion for this particular community. When it comes to the lung and the sample preparation, this is putting on your aerosolization hat and becoming an aerosol physicist again. It's being able to aerosolize the vapors or the particles in the right size regime for inhalation into the system. And so usually for OECD type guidelines, whenever you're trying to follow 90-day inhalations, you want to be between 1 and 3 micrometers in your aerodynamic size distribution. So we must make sure that we're aerosolizing these materials, but then the engineering question is, how then do you expose that aerosol cloud to the cells in culture? So you use settling chambers for gravitational deposition, or you can use gentle impaction. And so you can imagine, I'm not going to use my slides, but you can imagine a fairly larger system that needs to be created. It doesn't mean that we can't still use the microfluidics, but we have these chambers that we've built that will produce the cloud or the gentle impaction, and you insert in a drawer your chamber where the cells are housed underneath that for the exposure itself. And then you can take that chamber back out of the exposure system and then plug it back up into however you want to incubate it or do your endpoint analysis. So for both gut and lung, sample preparation is going to be very important, and thus the microfluidic systems or the 3D systems need to be modular enough to be able to use them in different scenarios for the exposures. And like Ed already mentioned, the metabolites itself. So if we know, we need to have the chemist involved as well to be able to digest or react with reactive oxygen species or NOx or ozone or any of the other materials or redox redox substances that we already heard about earlier today, we need to be exposing and doing our validation of the studies on the material itself, but also maybe metabolites or reacted materials. The third issue that we've kind of come across in the last couple of years is really thinking physiologically about the cells and thinking about, of course, the ratio of the numbers of cells, how many macrophages per epithelial cells you may want in your system. So is it a 1-to-4 ratio, a 1-to-10 ratio, a 1-to-100 ratio? That's important to consider what ratios you want to do. And then understand, characterize that, get the basal expressions of whatever ratio you thought about doing. But also the order in which you then drop in your, for lung, for instance, polymorph neutrophils or your dendritic cells. So if we're talking about cells that are recruited after injury, the system, again, has to be modular enough that whatever is being exposed, when we're talking about the healthy tissue that may be being exposed, that can be exposed, right? But not with the cells that are then recruited afterwards after the injury. So we have to be able to take the system and then add in other types of cells that might be there in an organismal level in which recruited cells would get to that point after the insult, after the injury. So making it modular like that. I still don't know how to deal with clearance after, so exposure to particles or vapors or whatnot, and then the clearance of those substances afterwards. I'd like to hear perspectives on how people are dealing with clearance afterwards, which will help us with the downstream effects, especially for those groups that are linking up their microfluidic systems to other organs in the human on a chip scenario. And the last big thing is dosimetry. So really including those toxicologists and other exposure people who are really thinking about the dose. Do we use the same dose? Is there going to, either we need to have the conversation with those mathematicians in the audience to make sure we're doing the right doses, because we have some really great models that have been developed for 2-dimensional monolayers, but do those need to be reevaluated and relooked at when we're talking about 3D or 4D type models? Other experimental designs, such as when we do these types of things, this is what I look at when I review manuscripts that have been submitted, is did they also study and do their experiments on the mixed cell population as well as the cells separately? And do we have to do that, or can we get away from doing that? So those are questions we think about, and I think I'm going to stop there.